Welcome to Biotanemy. Plasmid DNA isolation. Isolation of plasmid DNA is as much easier as genomic DNA isolation. All you have to know is about the 3-lysis solution you add to the cells to open it up and release the plasmid DNA. Now, let's look into the 3 solutions used in the plasmid DNA isolation. Solution 1. Alkaline lysis buffer solution. Solution 1 contains 50 millimolar glucose, 25 millimolar tris HCl of pH 8, and finally 10 millimolar EDTA of pH 8. The glucose will prevent the immediate osmotic lysis of bacteria and the shearing of DNA. EDTA, on the other hand, will disrupt the outer membrane of the plasmid by chelating the calcium and magnesium, which will allow the lysosome to enter and inhibit the cation-dependent nuclease activity, respectively. Note: the calculations for the amount that should be taken for each chemical will be given on the following website. Find the link given in the description box. Solution 2. Or lysis solution 2. Lysis solution 2 consists of 0.2 normal sodium hydroxide and 1% of SDS solution. The SDS will solubilize the phospholipid and protein components of the cell membrane, which will lead to the lysis and release of the cell contents. The addition of sodium hydroxide will denature both chromosomal and plasmid DNAs, along with proteins. However, optimized lysis time will allow the maximum release of plasmid DNA from the cell without the release of cell wall-bound chromosomal DNA, while minimizing the exposure of the plasmid to denaturing conditions. Make sure that you prepare solution 2 freshly at the time of plasmid DNA isolation. Solution 3. Solution 3 is a mixture of 5 molar potassium acetate, glacial acetic acid, and distilled water, in a ratio of 12 to 23 to 5.7. The potassium acetate will form a complex with SDS, protein, and genomic DNA to precipitate. The acetic acid will neutralize the pH, allowing the plasmid DNA strand to renature. Now that all the reagents necessary for plasmid DNA isolation are ready, let's move on to the procedure. Transfer about 2 milliliters of the cells that contain plasmid into an Eppendorf tube. Collect the cell pellets by centrifuging them at 10,000 RPM for 10 minutes. Add 10 microliter of ice-cold solution 1 into it. Vortex it thoroughly to homogenize the solution and incubate it over the ice for 5 minutes. Add 200 microliter of freshly prepared solution 2 to the mixture, mix well, and again incubate it on the ice for 10 minutes. After 10 minutes, add 150 microliter of solution 3 and continue to incubate on ice for 15 minutes. Finally, centrifuge the mixture at 12,000 RPM for 10 minutes. Collect the supernatant. Onto the supernatant add an equal volume of phenol chloroform and once more incubate it at ice for 15 minutes. After incubation, centrifuges the solution at 12,000 RPM for 10 minutes. Collect the supernatant and add twice the volume of 100% alcohol. Mix the solution and incubate it on ice for 30 minutes. Then, centrifuge the mixture at 12,000 RPM for 15 minutes to collect the pellet. Add 70% ethanol into it. Mix well, and centrifuge the mixture for 15 minutes at 12,000 RPM. Finally, discard the supernatant and air dry the pellets. Again, redissolve the pellets in 1.5 milliliters TE buffer of pH 8. That's all for today's video. Thank you for listening. Hope all of you understand the concept, and for more queries and proposal, please comment down. See you all in a new topic.